Welcome back to our fourth and final video for Chapter 8. At the end of the previous video, we were discussing photosynthesis, and we mentioned light-dependent and light-independent reactions. Let's look more closely at those light-dependent reactions of photosynthesis. So photophosphorylation is the process of making ATP from ADP and phosphate using energy from light. There are two types of photophosphorylation. Non-cyclic uses photosystems 1 and 2 and produces ATP and NADPH, in addition to oxygen if it's oxygenic photosynthesis. This is sometimes also known as the Z scheme because of how it's usually diagrammed, and I'll show you that shortly. The second type of photophosphorylation is cyclic photophosphorylation. This one uses photosystem 1 only, and it makes extra ATP, but no NADPH or oxygen. Cyanobacteria and chloroplasts can switch between both of these modes, non-cyclic and cyclic, whereas in anoxygenic bacteria, they only use photosystem 1, so they only have cyclic photophosphorylation. So here is non-cyclic photophosphorylation, and let's start at the diagram down here. It uses both photosystem 2 and photosystem 1. So light will excite the pigments in this photosystem 2 antenna pigment system, and then eventually the reaction center will lose its electron, and its electron will travel through the electron transport system. This will generate a proton motive force to generate ATP. Meanwhile, that electron will be donated to the second reaction center that is part of photosystem 1. The electron is excited by more light, and eventually the electron will be donated to NADP+ to form NADPH. So for oxygenic photosynthesis, water will be split at PS2 or photosystem 2 to provide replacement electrons and releases oxygen as a product. And I can kind of see that a little bit better here. So light excites the electron at photosystem 2. That electron passes through my electron transport system, enters photosystem 1, and eventually that electron is used to produce NADPH. Meanwhile, the proton motive force generated by the electron transport system here allows protons to generate a gradient such that they want to move back down the gradient, and that will form ATP. In contrast, in cyclic photophosphorylation, only photosystem 1 is involved. So light will excite those electrons. The electron will cycle through the electron transport system. Remember, that generates a proton motive force to generate ATP. And the electron simply just comes back and replaces the previous electron that was excited. So it's the same electron being cycled over and over again. No NADPH is generated. No oxygen is needed to replenish the electron here. So this is used when ATP's demand is higher than NADPH demand. And a key contrast is cyclic, produces ATP only, versus non-cyclic photophosphorylation generates ATP, NADPH, and oxygen if it's oxygenic. So why would a photosynthetic bacterium have different pigments? Different pigments will absorb light at different wavelengths, and this will allow bacteria to capture light energy in environments where sunlight is filtered or maybe limited, like deep in the water or in shaded areas. And this allows them to avoid competition by using wavelengths that might not be absorbed by other organisms, and also maximize energy efficiency by covering more of that light spectrum. So for example, I mentioned chlorophylls absorb mostly blue and red light. Carotenoids absorb in the blue-green region, so they appear kind of reddish-orange. There's something I didn't mention, like phycobilins. Those are actually pigments that absorb green, and they're useful in cyanobacteria in aquatic habitats. So in short, different pigments will really expand the usable light spectrum and help microbes adapt to diverse environments. Now let's look more closely at the light-independent reactions. These are also known as the Calvin cycle or the Calvin-Benson cycle, named after the two scientists who discovered the cycle. There are three phases to the light-independent reactions: fixation, reduction, and regeneration. First is fixation, and in fixation, carbon dioxide combines with RUBP, which stands for ribulose bisphosphate, and this is catalyzed by the enzyme Rubisco. Rubisco, you don't have to know it, but it stands for ribulose 1,5-bisphosphate carboxylase oxygenase, and it's widely considered one of the most abundant enzymes on Earth. This will form 3-PGA, or 3-phosphoglycerate. In the next step, reduction, ATP and NADPH will be used. Remember, these came from those light-dependent reactions. Those will be used to reduce 3-PGA to form G3P, glyceraldehyde 3-phosphate, which we saw before in glycolysis. G3P can be used to make glucose. Finally, in regeneration, some of the G3P that's not used to make glucose will be used instead to regenerate ribulose bisphosphate so the cycle can continue. And the Calvin cycle will occur in the cytoplasm of prokaryotes like bacteria, because remember, they don't have chloroplasts. And in eukaryotes that do have chloroplasts, the Calvin cycle will happen in the stroma, that liquid portion of the chloroplast. So the three stages of the Calvin cycle include 1, fixation. Rubisco is going to take CO2 plus RUBP to form 3-PGA. In the second stage, reduction, ATP and NADPH from those light-dependent reactions will be used to reduce 3-PGA and form glyceraldehyde 3-phosphate, or G3P, which is going to be used to make glucose. And finally, in the third stage, regeneration, some of that G3P will be used to regenerate ribulose bisphosphate so that Calvin cycle can continue. All right, let's recap 8.6. For the first one, describe the function and locations of photosynthetic pigments in eukaryotes and prokaryotes. So remember, the function is to absorb light energy at specific wavelengths. This will excite electrons and drive ATP and NADPH production. In eukaryotes, the pigments are located in the thylakoid membranes of chloroplasts. In prokaryotes, the pigments will be embedded in the enfolded plasma membrane since they don't have chloroplasts. For the second one, major products of light-dependent and light-independent reactions. For the light-dependent reactions, these were ATP, NADPH, and oxygen if it's oxygenic. For the light-independent reactions, or the Calvin cycle, we generate glucose or other carbohydrates, and we regenerate RUBP so the cycle continue. For the third one, describe reactions that produce glucose in a photosynthetic cell. So this is in the Calvin cycle. We had fixation, where Rubisco will take CO2 and fix it to form 3-PGA. In the second one, reduction, 3-PGA is used to form glyceraldehyde 3-phosphate, using up that ATP and NADPH from earlier. And in regeneration, we're going to regenerate RUBP so the cycle continues. So some G3P is going to exit that Calvin cycle and be used to form glucose and other sugars. Finally, compare and contrast cyclic and non-cyclic photophosphorylation. In non-cyclic, which is also called the Z scheme, it uses photosystems 2 and 1, produces ATP, NADPH, and oxygen. And in cyclic, we're only using photosystem 1, and it produces only ATP. The electrons will continue to cycle back to photosystem 1 instead of reducing NADP+. So in cyclic and non-cyclic, both of them make ATP, but only non-cyclic also produces NADPH, as well as oxygen if it's oxygenic. Now in 8.7, we're going to look at biogeochemical cycles. So in this section, we're going to focus on how microorganisms move elements like carbon, nitrogen, and sulfur between living things and the environment. And by the end of this section, you should be able to explain why microbes are essential in these cycles and give an example of bioremediation. Energy flows one way, from sunlight to heat, but matter cycles. And microbes are nature's recyclers, breaking down organic matter and converting it back to forms that other organisms can use. They are critical for the cycling of elements like carbon, nitrogen, sulfur, and many others. Let's start with the carbon cycle. These organisms are involved in the carbon cycle, including autotrophs like plants, algae, and many microbes. These will fix carbon dioxide gas into organic compounds through photosynthesis. Heterotrophs, including humans and many bacteria, do the opposite. They break down organic molecules and release CO2, carbon dioxide, back into the environment through respiration or fermentation. For the next one, in anaerobic environments, methanogens produce methane gas, while the last one, methanotrophs, consume methane and help reduce greenhouse gas levels by reducing gases like methane. We said autotrophs fix carbon dioxide, so they're going to turn it into an organic compound that heterotrophs can use. Heterotrophs will then break down these organic compounds and release CO2. We also talked about methane gas. So methanogens produce methane in anaerobic environments, most likely, or most of the time, and methanotrophs will consume methane and reduce greenhouse gas levels. Looking at the interaction between heterotrophs and autotrophs in the carbon cycle, autotrophs like plants, cyanobacteria, or algae, they can fix inorganic carbon, CO2, into organic molecules through photosynthesis. Some can undergo something called chemosynthesis, which is when you make organic carbon using energy from inorganic chemicals. Heterotrophs, on the other hand, like animals, fungi, or most bacteria, will consume these organic molecules for energy and carbon and release CO2 back into the atmosphere through respiration. And then that CO2 can be reused by those autotrophs. So this reciprocal exchange really maintains balance in the global carbon cycle. The nitrogen cycle also relies heavily on microbes. In nitrogen fixation, by species like Rhizobium or cyanobacteria, they're going to convert nitrogen gas into ammonia, NH3, or ammonium, NH4+. Ammonification by bacteria and fungi can turn organic nitrogen waste into ammonia. Nitrification by Nitrous ammonius or Nitrobacter oxidizes ammonia to nitrite and then to nitrate. Finally, denitrification by bacteria like Pseudomonas or Clostridium converts nitrate back to nitrogen gas, completing the cycle. Many biological macromolecules include nitrogen, such as proteins and nucleic acids, but getting nitrogen into living organisms is pretty difficult. So luckily, we have all of these microbes involved in the nitrogen cycle. They can fix nitrogen for us, converting it to ammonia or ammonium, nitrite, nitrate, etc. And all of these products can be used by organisms such as humans to build their proteins and nucleic acids. In the nitrogen cycle, we saw nitrogen fixation, which is converting nitrogen gas into ammonia or ammonium by microbes such as Rhizobium. For the second one, we saw nitrification, which is the conversion of ammonia into nitrite and nitrate by nitrifying bacteria like Nitrosomonas or Nitrobacter. We also had denitrification, which is the conversion of nitrate to nitrogen gas by denitrifying bacteria such as Pseudomonas or Clostridium, and also ammonification, which is the decomposition of organic nitrogen, and that converts it into ammonia, and this can be carried out by many decomposers like different microbes or fungi. In the sulfur cycle, sulfur oxidizing microbes convert hydrogen sulfide into elemental sulfur and then sulfate. Under anaerobic conditions, sulfate-reducing bacteria do the reverse, producing hydrogen sulfide. Finally, decomposer microbes also release sulfur. They can break down organisms, releasing sulfur from amino acids and return it to the environment. So for the sulfur cycle, we have sulfur oxidizing bacteria that convert hydrogen sulfide into elemental sulfur and then sulfate. We have sulfate-reducing bacteria that do the opposite, take sulfate and convert it back into hydrogen sulfide. This is usually in anaerobic environments. And then we have our decomposers that release sulfur from amino acids back to the environment. Bioremediation is the use of microbes to degrade pollutants such as oil, solvents, or synthetic chemicals. In situ, bioremediation treats contamination on site, which is cheaper but harder to control. Whereas ex situ, bioremediation removes the contaminated material to treat it elsewhere under optimized conditions. Enhanced methods add nutrients or oxygen to encourage the growth of pollution-degrading microbes, or sometimes specialized microbes are added to speed up cleanup. Let's compare and contrast the benefits of in situ and ex situ bioremediation. In situ is performed at the contamination site, so microbes are used to degrade pollutants directly in the soil or in the groundwater. The benefits is it's less disruptive, lower cost, and treats contaminants in that place, but the limitations are it's harder to control environmental conditions like the oxygen, pH, and other nutrients or other variables at that site. Ex situ bioremediation is when contaminated material is removed and treated elsewhere, like in bioreactors or land farming. The benefits are the conditions can be optimized. You control the environment, like the temperature, aeration, the strains of microbes in that region. The limitations are it's more expensive, right? It requires excavation and transport, and it might not be feasible for larger sites. All right, let's recap 8.7. Describe the importance of microbes in biogeochemical cycles of carbon, nitrogen, and sulfur. For the carbon cycle, we saw autotrophs fix carbon dioxide into organic matter. Heterotrophs break down those organics and release CO2. Methanogens produce methane, and methanotrophs consume methane. In the nitrogen cycle, we saw microbes fix nitrogen gas. They can decompose organic nitrogen, nitrify ammonia to nitrate, and denitrify nitrate back to nitrogen gas, making nitrogen available in many different types of usable forms. In the sulfur cycle, we saw sulfur oxidizing bacteria convert hydrogen sulfide to sulfate, and sulfate reducing bacteria do the opposite, reduce sulfate back to hydrogen sulfide. We also saw decomposers recycle sulfur from amino acids. So the importance is that these microbes are essential because they drive the chemical conversions that recycle these elements and keep our ecosystems balanced. For the second one, bioremediation. So bioremediation was the use of microorganisms to degrade or detoxify pollutants in the environment. So an example could be oil spill cleanup using hydrocarbon degrading bacteria. Another example could be we can actually use Pseudomonas species to break down solvents or industrial contaminants as well. All right, and that wraps up Chapter 8. In Chapter 9, we're going to look at microbial growth. Thanks for watching.